Hi there. In this video, we're going to see how you utilize other carbon sources in our food. Um, all of them are sugars. The next two that we saw galactose. Of course, we've seen glucose. Fructose and is of course a sugar. Glycerol is not, but as we have seen already in gluconeogenesis, it can be used to enter the glycolytic pathway and either make glucose, as we saw in gluconeogenesis, or actually to be consumed through glycolysis. So let's start with mannose. Is again another isomer of glucose, nepimer. So we have now in this case we don't have carbon 4, we have carbon. Let's see which one now. Carbon 2. That's different with mannose. Right here. Oh, hold on, let me find my pointer. Okay, right there. So mannose is actually fairly easy to utilize. It's gonna get phosphorylated by hexokinase. It's a hexose, so hexokinase will see it. It will phosphorylate it at the 6th position, and then can be isomerized to fructose 6-phosphate. The isomerization is exactly the same when we had seen from glucose and fructose, going from glucose 6-phosphate to fructose 6-phosphate, the second enzyme of glycolysis. It's exactly the same reaction. Mechanism, so instead of having glucose 6-phosphate, what you have here is mannose 6-phosphate. By the way, mannose is found in several fruits, and then we're done, right? I mean, we got fructose 6-phosphate, and it can go down through glycolysis. Enter, we have the PFK step here, and so on and so forth. Before we return to fructose, which is another sugar of huge significance and is actually a lot in the news, let's just review again, remind everyone how glycerol gets utilized and enters glycolysis. Again, glycerol is not a sugar, okay? You need a carbonyl somewhere, and we don't have it. We do have oxygen on all 3 carbons, but they are all hydroxy carbons. Nonetheless, because it's a carbon fuel used quite a bit, actually, then we need to discuss it again. So first it gets phosphorylated to glycerol 3-phosphate, and then here is our carbonyl now. So this hydroxy carbon on position 2 gets oxidized to a ketocarbon, and very simple reaction, and that's it. We're done. So that's dihydroxyacetone phosphate, and then it enters glycolysis. Now let's talk about fructose. Fructose has, as you see here, 2 entry points. Okay. It can enter as fructose at the fructose 6-phosphate position, or it can be broken down and enter really at the bottom of the upper part of glycolysis as at the aldolase step as a dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So this has very important implications. If you enter at this point, okay, you bypass PFK, which is a major control point, right, to regulate glycolysis. So let's look at the reactions in a little more detail. So that's a straight up introduction, which is used in most tissues, but not in the liver. Significantly, that's a very significant distinction. The liver does not do it. In all other tissues, what happens is fructose gets picked up by hexokinase, and it gets phosphorylated at the 6th position. So you got fructose 6-phosphate, and then you're done. Okay, so you continue on with PFK and so on and so forth. But the liver doesn't do it. Instead, the liver does this part. So it enters at the aldolase step. And what happens here is fructose gets phosphorylated at the 1 position. So you have a fructose 1-phosphate. There's an enzyme called fructokinase. Much as we had seen with galactokinase, which phosphorylates galactose at the 1 position, there is a fructokinase that puts a phosphate at the 1 position. Then you have a special aldolase, fructose 1-phosphate aldolase, and what you get from that, it works in the same way. It's an aldolase, so it cuts it in half, and you get dihydroxyacetone phosphate. That's one. Part that's straight into glycolysis, and then you have glyceraldehyde. So as long as you phosphorylate that, and there is a triose kinase, as there is a hexokinase for 6 carbons, 6 carbon sugars, there is a triose kinase, and it will pick up glyceraldehyde and phosphorylate it and become glyceraldehyde 3-phosphate, and now you end up with the two trioses that are ready to go in the lower part of glycolysis. The significance of that for human health is that in all these high fructose content drinks and other foods that are sold today, um, so a lot of the fructose that we metabolize goes through this pathway. And when that pathway is operational in the liver, you lose the control at PFK. So you keep breaking it down, and eventually you put it into fat or whatever. And so it's a huge problem. Right? And that's the reason. All right, so that we describe the major sugars, how they're utilized: galactose, mannose, fructose, and now we're going to move in with the next videos on how we make glycogen. This is the sugar reserves that we have in our cells, and how we break it. But that's a topic for the next videos. So we'll stop here.